This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 991, recorded on March 10th, 2023. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today here in New York at the incubator, Dixon Despommier.
Hello, Vincent, and everybody else. Why
do you insist on saying hello, Vincent, and everyone else when there's no one else here yet?
Everybody knows there's going to be somebody else now. Come on.
Well, if there weren't, would you not say and everybody else?
Oh, that's right. I wouldn't. If it was just the two of us, I would start an enormously long philosophical discourse.
Okay. What you were going to say, the weather or something?
I was going to say, I would love to tell you what the weather's like outside, but we don't have windows here, so that's not going to happen.
Well, it's 8 degrees Celsius and light rain.
You believe all you read?
No, it could be wrong, but you're right. When you came, which was about an hour ago, was it raining?
It was no, it was fine.
Also
Absolutely.
Joining us from Austin, Texas, Rich Condit.
Hi, everybody.
Don't tell us it's 83 degrees and beautiful.
No, but it's 71 degrees and beautiful. 71 degrees and sunny, and we're looking at similar stuff in the future. The bluebonnets are up.
Oh, great.
The red bud has peaked and is on its way out. The leaves are coming out on the trees. The grass is growing.
Oh, I love this.
The bees are buzzing. The birds are singing. The irises are up. Man, it's awesome.
Wow. Here it's still winter, right?
It's still.
But you can tell it's kind of trying to creep out of it.
The trees are getting fat. I told that to my wife as she was.
Did they really?
Yeah, they are. You can actually see the difference.
What is it, like 2 or 3% swelling?
Yeah, but if you're looking through a bunch of them, you can't see the end of that.
I see. I got it. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 49 Fahrenheit, 9 Celsius, and it's clouding over. It was sunny, and now it's more and more cloudy, and we're supposed to get
becoming ominous.
Supposed to get some slush this evening.
That's
some
right.
rain, snow, alternating. Yeah.
If you have never been to Texas this time of year, when the bluebonnets are blooming, it is something to behold.
Yes.
It is a phenomenon. There are carpets of these things all over the place.
Wow,
It's really,
cool.
it's
Are
really
they
pretty cool.
related to lupin?
Is that why you're
by
wearing
any
a blue
chance?
shirt?
Yeah, well, no, that's not why I'm wearing the blue shirt, but good point. I can't tell you in fact, Dixon, but it certainly looks it.
Before we hop into our science, 2 announcements. ASV 2023, American Society for Virology, Athens, Georgia, annual meeting, June 24th to 28th. Go to asv.org/ASV 2023 for more information. Members can apply for support for dependent care during the meeting, ASV Cares, or you can sign up on the meeting site for on-site child care through Kitty Corp. Go to asv.org/ASV2023/childcare. We have 1,000. We have some tickets left, like not a lot, maybe 10.
Oh, wow.
So Vincent at microbe.tv if you would like to come. I know I'm going to oversell because I can't keep count, and people are going to be yelling at me.
We'll just have SRO, that's all.
Did you save a couple extra seats for our guests, for like people who are accompanying us?
I got you down. I put you on the list, yeah. And
some comps.
Angela put
Yes.
one down, but nobody else. Rich said someone might, but no one has.
I'm solo.
It's just solo. Dixon is solo. Kathy's solo. Brienne is solo. So.
And of course, Hans is solo.
Yes, always.
And so it's shaping up to be a nice event. I am, as you know, we have the Randy Despommier Quartet.
You do.
They'll be playing before and after. There's a little nook to the right of the stage. So, Rich, what are the stage directions? Facing the stage or facing the audience?
Wait a minute. I blanked out there for a minute.
When
What do you wanna know?
when you're standing on the stage facing the audience, stage right is to your right.
Yeah.
That would be
That's
house
the way you
left.
give directions. You don't
Yes.
stand in the audience.
Stage right or house left.
Oh, you could
Yeah.
be house left would be facing the stage.
House
Yeah.
left and stage right are the same thing.
Oh, cool. Anyway, house right, there's a little alcove where the quartet is going to sit. And they will play before as you're seating. They will play afterwards as you're leaving. Although now I am working on leasing upstairs. So upstairs from the theater is a very nice reception area with a bar. And I can lease that and have everyone go upstairs and mingle with us.
That would be easier.
And then the quartet could come upstairs and play there.
Oh,
So
that would be great.
yeah, we don't have to walk anywhere. The previous idea was to walk to the incubator, but there's not enough room for 130 people here.
Got it.
Right, Alan? You've been here.
No, that would not be a good scene. I mean, back when I was in college and grad school, we would have had a party of 130 in a space that size, but it would have been like, stand with your drink on your
you'd put it in
shoulder.
a hall.
Yeah.
You would be right next to each other, and when you raise your drink, you'd spill
And if
it
the
on
fire
somebody.
marshal showed up, that's the end.
Oh,
of it.
that's the end
That
of
would
the
be
day.
the end. That's right.
So I'm going to probably book that upstairs space for another hour and a half.
That would be beautiful.
And there will be bartenders, and you can chat with us, and the band will be playing.
Will there be some food?
Yeah. I think they will have some, hors d'oeuvre type thingies, right?
Okay.
I'm gonna, I'll make sure I can't bring in my own food. It has to be provided by
Right,
them
of
because they
course.
don't want to be responsible for me poisoning everyone. You know?
Exactly. They want to be responsible for
They
poison.
want to
they-
be, yeah. They
Well, they have insurance. I don't have insurance.
Right.
Okay, so that's TWiV 1000. Vincent at microbe.tv if you would like tickets. Okay, and we are suggesting a $100 donation to defray the cost of the theater, which the theater and the band, if we sold all the tickets, it would cover it.
Wow.
So we would appreciate your help. That way we don't have to dig into production money to do this. But this is a once-in-a-lifetime thing. It's cool that we reached 1,000.
It's amazing.
A little nerdy podcast, reaching 1,000 episodes, and a lot of people grateful. So we're going to celebrate it. It's just going to be us, folks. Just us. There's no special guest. Tony Fauci's not
Hey,
coming.
we are special guests now.
Nobody
Come on.
else is coming.
Well, if Tony buys his ticket, he's welcome to come, but he's not going to
Absolutely.
be like featured
Absolutely.
on stage.
So it's funny. The theater has a VIP entrance, right? It's separate. Where she said the limo can pull up and the VIP could go slip right in the door and go behind the stage right into the green room, right? And she said, if you want, you can have their security vet the place beforehand.
That's funny. That's funny.
She said, we have some celebrities
How
coming.
much is it?
Okay,
cost
well,
for a
I'll
red
make sure my
carpet?
security vets the place, and I'll,
Okay, you
I'll
send
tell
them
the
down,
limo
Alan,
driver
yeah.
to pull up out back. Yeah.
You want a red carpet, Dixon?
Well, what the heck?
All right, so that's TWiV 1000. And now we're going to do some papers. But first, I got a letter this week from Jeff, who is a cardiologist in Columbus, Ohio. And I took this very seriously. He said, I was watching TWiV 989 last night, which was the last Friday episode, which I thought we had a nice chat,
You
right?
did.
But he said, when you and your group are discussing some of the papers, I don't understand much of the discussion. If you want a more general audience, then I think it would be best to speak to that audience and define for us non-virologists what you're talking about. I know this takes more time, but I think it will keep your audience larger and better informed. I find your topics fascinating, but there are many times I'm completely lost in the jargon. Thanks again. So let's keep this in mind. I'm going to try and explain everything, and then when we're done recording tomorrow night, we'll see if it worked.
And Jeff, I mean, we do actually try. We've got little reminders in the show notes at the top. Remember, or Caleb and Cherish are listening, which is a couple of people who used to be young listeners and are probably like, having their own kids now. But yeah, this is something that we need reminders of, I think,
Oh,
periodically.
yes, because we slip
it's
into jargon, because
so
we're scientists. and
easy to forget
That's
that
what we
not
do.
everybody knows these terms.
I have to admit, it's fun to slip into jargon amongst ourselves, right?
Well, because
tell me
that's
the cardiologist-
purpose. Yeah, the purpose of the jargon is not to obfuscate. It is to make it easier for us to communicate about these concepts. But
Right.
if you don't know it, then it forms a barrier.
So I was teaching about MHC the other day in my class, and I said the peptide is loaded onto MHC, and I stopped and I said, that's jargon, isn't it?
That's jargon. That's right.
Right? It's loaded. I mean, I guess you load a truck, so maybe people would get that, right?
Yeah.
I dare say, though, that at the cardiology meeting that Jeff goes to,
I'm sure there's jargon.
There's an S-load of jargon.
Yes.
All right, so we will try and explain things and if one of us uses jargon, please someone else say, hey, this is what that means.
We'll try
I'm
to-
usually
try to
the one
police
that
each
says it because I'm not a jargonist with your.
The jargonist.
No,
Jargonist.
I have my own jargon for my own subject, but I
Okay.
thought I was the voice of the people, basically.
You're a
And,
scientist.
I am a scientist, though. That's
Well,
true.
you're talking about field-specific
Yeah,
jargon.
exactly.
All right, so give me an example of parasite-specific jargon.
You could start talking about stages of nematode development and molting of cuticles and first stage and second stage.
It's not jargon. That's the accurate
No,
terminology.
I know that, but people might not know what a stage is.
Isn't that stage left and right? Okay. Anyway, we're going to do this. So the first paper, Journal of Virology. Pandemic 1918 influenza virus does not cause lethal infection in rhesus or cynomolgus macaques. I picked this because I wanted to emphasize how animal models can be so different than humans in responding to viruses, right? And so I'm just about to do the virulence part of my course where I say, you know, virulence is very tricky to measure. So this paper was published in the Journal of Virology. We have two co-first authors, Mabel Chan and Meenakshi Tiwari. And the last author is Darwin Kobasa. And this comes from the Public Health Agency of Canada, from Oregon Health and Science University, from the National Institutes of Health up in Hamilton, Montana, and the University of Manitoba. So as many of you know, the 1918 influenza pandemic was a big one. Killed 50 to 100 people, 100 million people, 50 million to 100 million people worldwide, which is more than COVID so far. Correct, everyone?
I
Correct.
think that's
Yes.
correct. And the population of the world was smaller then by a large margin. So this, when you look at demographics and life expectancy for countries around the world, you see a dip in life expectancy
Yeah.
in the graph of all of history because of 1918 flu. It was a huge, huge thing.
So,
So-
Vincent, can you explain why there's this big difference between 50 and 100 million? How could you make such a big error?
How could you be off by 50 million?
Well, because there was no WHO then, or was there?
I would say it doesn't matter because either way, it's hard to know what people die of, right?
Yeah, but we have
Yeah,
much
that was-
better ways of knowing now than we did then.
Yeah, we
Those-
didn't even we had very what we would now consider very poor record keeping of
Correct.
just people, let alone health. And it was hard to know how many people died of what.
Do you
This
think
was
that's
also complicated by the fact that there was a global war going
Yes.
World War
right.
One,
on, which
yeah.
not only were people actually actively killing each other, but the conditions of the war contributed to a lot of mortality as well.
Yes.
But I think even today, you know, when someone dies and COVID was involved, sometimes it's not put on the death certificate that it was COVID, right? It would be a myocardial infarction or whatever, stroke. So it's hard.
Conversely, sometimes when people die and they had tested positive with COVID, they may not have died of COVID.
That's right.
Yeah.
That's right. So anyway, the why this virus killed so many people is still not entirely known. I mean, because the ability of viruses to kill is based on the virus properties and also on people, and also on healthcare. Right? 1918 wasn't great healthcare, and so probably that was a factor as well. So what you try and do is have animal models to study these viruses and try and get some hints about why it might be more virulent. So, and by the way, virulence in virology is the
right.
capacity of a virus to cause disease. We have an avirulent virus, which causes little or no disease. And if you make attenuated vaccine strains, you hope they're avirulent. And then you have virulent viruses that kill you and everything in between. And so this 1918 influenza virus is highly virulent in mice and ferrets. When you give them 1 million, 50% tissue culture infective doses. a TCID50 is the amount of virus you need to infect 50% of your targets. If you had 100 wells, cell culture wells, to infect 50% of them, or 50, that's how much virus you would need. That's how we measure it. So 1 million TCID50 is a lot of virus.
Importantly, it's a quantitative measure of infectious virus.
That's right.
Yes.
I mean,
and so you can do these comparisons with different flu viruses, which vary dramatically in their level of virulence.
That's right.
And so 1918 flu was an especially virulent flu. And so a lot of these models are trying to see, does your mouse model or your ferret model represent that? If you infect them with a less virulent flu, do you get less death in the same dosage? So in this paper, they're looking at macaques, which are a major primate model, a monkey major primate model for studying this, that are thought to be a good model of human disease.
By the way, the if you wanted to compare pandemic influenza viruses from 1918, 57, 68, 2009, you'd have to do them all in one study, really.
Yes.
Because you can't do one of them and then go into the literature and compare it, because likely they use different approaches, and that really makes it difficult to compare. So, they say that mice and ferrets, they're good, but they do not recapitulate human disease pathology, and therefore it's hard to test human-specific therapeutics, right? What works in a mouse may not work in people. And we would like to have therapeutics for 1918 in case it should ever return, but
or for a flu virus of equivalent lethality.
So, again, if I were not knowing anything about this, but I knew about the flu virus, what other animals does the flu virus ordinarily infect?
In nature, naturally,
Mm-hmm.
well, they infect birds of all kinds, right?
Yeah,
Water
that's
birds.
avian influenza, though, right?
Yeah, but human strains can go back into them as well, and they can infect pigs in the wild,
What
and
about
horses, dogs, right?
other primates?
I don't think that non-human primates are hosts for influenza viruses in nature. I'm thinking of a picture I have in my head in a review article where I show all the hosts of flu viruses, and I don't see a non-human primate there. I see a lot of birds and pigs and
So I'm
dogs
wondering,
and horses.
why would they pick these two non-human primates as their...
That's a good question, because they're
Thank you.
99% genetically identical to humans,
Yeah, but
right?
you can find even closer than that if you use chimpanzees.
Well, they're about 99%. So probably the macaques and the cynomolgus are less than
But
99%.
How
right?
many primates are there? Right.
That's out of my lane.
But you can look it up on Google. It's a lot. We have a lot of primates
And-
out there. So why not try them all? And if you don't...
Well, because you have to be able to breed them, right?
I got it.
And so right now, no more, we can't use chimps. They're outlawed,
Correct.
right? Which is great.
And even if they weren't, they are extremely expensive and difficult to work with.
And they're really cute.
Yeah, but it
and they're
sounds a
cute,
little
and
bit like the joke where the guy is looking for his watch, right? And,
right under the lamp, because
under the
that's where the light
wrong-
is.
Yeah, because that's
yeah,
where the light
because
is.
the light's
yeah.
better. But I'm curious as to why
Well,
they...
listen, they say here non-human primates are important for testing treatments in humans. I mean, I don't know if that
Treatments,
may
yes.
be true for influenza virus, but it's not true for all
But
viruses.
lethal, they're using the word lethal here because they're using death as an endpoint.
Yeah, they
Right.
would like
So
to
what you would like to be able to do, I mean, most people, even if they're against animal research, they don't want to necessarily take a drug that hasn't been tested in
That's
anything
right.
else, right? I mean, you want the bugs worked out before you take it.
Here.
But
And so we want to have a good model of flu infection that represents the pathogenesis, that represents the virulence of the virus. And cynomolgus macaques or other macaques are a pretty good candidate. They're very closely related to us. You can work on them. They're not as expensive or difficult as chimpanzees. So it looks like they ought to be a good model for this.
So, Rich, when they were testing the smallpox antivirals, did they use a non-human primate model?
Yeah, that was part of the deal. They
Okay.
used just about every model you could get your hands on.
Because they couldn't do clinical trials in people, right?
Right. I mean, it was a big deal. This was I think the first successful application of what the FDA calls the animal rule.
Yeah.
Okay, where you have a situation, in this particular case, you're talking about smallpox, where there's no, you can't do an experiment in humans,
Yeah.
and you want to test the efficacy of a drug, so you have to use an animal model. What animal model are you going to use? And it was really complicated because you had a couple of different pox viruses to work with. You wouldn't necessarily. They did do some experiments with smallpox, but you don't. That's dicey because it's hazardous.
Yeah.
And smallpox in animals other than humans doesn't induce a disease that looks exactly like what it looks like in humans. So, you try stuff like monkeypox in primates, but that's not smallpox. All right. So you do everything you can.
So
And
is it-
in that particular case, the drug worked no matter what you did.
Right.
Okay. So that helped out.
It helps to allow the listener to understand the history of this approach if you go right back to Koch. Because Koch was a microbiologist who discovered the pathogenicity of cholera. And he did that by taking the bacteria and infecting other people with it. And then he infected some other animals, and they got the same disease as people. As a result, he said, this is what you have to do to prove an organism that's pathogenic for humans, it's pathogenic for this animal, and it's pathogenic for humans again. Koch's postulates require that you infect another animal and get the same result. But there are lots of exceptions, and I think we're looking at one of them right here.
So is it correct to say that if you want to get a antiviral or viral vaccine into humans, you need to have a non-human primate as part of the preclinical testing?
Okay. So
Yeah, right.
traditionally, the FDA has required
Two animals?
animal models, and for many things, they either require or really, really strongly urge you to do non-human primate experiments before you move into human trials and before they'll sign off on that. Very recently, the FDA had a rule change that they will actually allow people to proceed to clinical trials without animal data if you can show that your preclinical non-animal models are robust enough. And this is driven by tremendous amount of research that's been going on with things like organoids, which are combinations of cells in culture grown from stem cells that are like organs, and you can actually even string these together. So you can have a miniature liver, a miniature heart, miniature lungs, and you can put them together and do drug tests on these, so you don't have to use animals. Because to be frank, nobody wants to do animal experiments, because not only do you have to work with animals, which is kind of sad, but the bigger issue is that scientifically these are really not perfect models. They're not humans. They're not human cells. They don't have human physiology. It's just, this is what we had that we could use, and we would love to be able to have better models, and we are gradually moving toward a world where we do have better models. But in the meantime, we've got animal models. And so this paper, which we are going to get to, is asking the very simple question: if we put 1980, well, simple, but not necessarily easy to do, if we put 1918 flu into macaques, into these monkeys, how lethal is it? And so what's what we would call the lethal dose? 50%. So how much virus do you need to give? What dose do you need to give for half of the monkeys in your experiment to die?
So I
much the
remember the COVID mRNA vaccines were put into non-human primates. Right?
Yes.
So they went into mice for immunogenicity, and then they went into non-human primates, and then they went into people. So, all right. So here, I mean, part of the issue with non-human primates is, unlike inbred mice, they're not inbred.
No.
and they're typically colonies made from captured animals. So you go out, you capture some animals, males and females, and you breed them and you make a colony. And you use those. And so they're outbred, and each one is slightly different genetically, right?
Yes,
So
that's right.
that's the limitation. You have to do these things.
But that's more real life situation than it is.
It's
if you use
true.
an inbred.
Yeah, humans are outbred. Yeah.
Yeah, it is, but it also raises a statistical problem. I mean, yes, if
Of
you could
course.
do this experiment
Yeah.
on 10,000 monkeys,
right.
then you'd like to have the genetic diversity. But you can't afford to do that, and you don't have the time to do that. Nobody wants to do that. So you're really going to do the experiment on like 4 monkeys.
Yeah.
And you'd really like them to be as genetically close as possible to limit the variability across
Yeah.
those.
And then you'll see how that plays into this story too,
Yeah.
as well. Anyway, so first, rhesus macaques, they infect 4 of them, 4 male macaques with 7 times 10 to the 6, 7 million plaque-forming units. Is that jargon?
Yeah.
But
It is.
it's jargon we've kind of explained before on TWiV. Plaques
It's just
are these cell culture
Way to quantitate how much virus.
indications that you
Yeah,
get of
it's another
viruses.
quantitative measure of
Infectious virus.
Infectivity. Okay, a lot. It's
infectivity.
quite a bit. And get this. This is very interesting. They infect them intranasally, in the eye, orally, and intratracheally, because you don't know what's the best route. With 4 animals that are probably $30,000 each, you want to make sure you infect them. I once asked Vincent Munster, who's at Rocky Mountain Labs, why do you do all these routes? He says, to make sure they get infected, because we can only use 3 or 4.
So 1 root per monkey?
No, they get all.
No, all of them got all the
they
roots.
all got all 4 roots.
Yeah, they get all 4 roots.
Okay.
Because you don't really know which
I
is the
got you.
optimal route, and
Right.
we know that all of these may work in people. So, all right, then they follow these monkeys. They take bronchoalveolar lavage fluid, so they sample the lungs to see what virus is there, vital signs. They take temperature, and they monitor them for clinical disease. They weigh them. This is a good measure of severity of disease. All right, so these animals, minimal weight loss. Only one had an elevated respiratory rate that got better 2 days later. No signs of fever. No changes in heart rate.
They're fine.
They're fine. They really are very fine. Minimal. They say minimal clinical impact of 7 times 10 to the 6 PFU of 1918 influenza virus on rhesus macaques. By the way, this has to be done in BSO-4 lab.
Yeah.
Okay.
Which
Because
is
it's 1918
Actually,
flu.
I have a question that I could ask at the end, but I'm going to ask it
You can.
now.
Sure.
And it's partly for the audience and partly for me. 1918 was over 100 years ago. So
Yes.
It was.
where did this critter come from?
Well, that's a good point, because we didn't have the 1918 virus in 1918 because nobody knew what was influenza virus. But this came from Jeff Taubenberger's group and others who, in the 2000s or 90s, the Army kept paraffin blocks from people who died of this disease, right? The paraffin-embedded formalin-fixed tissues that you can slice very thinly. You look at them under a microscope. You can look at lung pathology, and they kept them. This is the Army. And so they went back to those blocks, and they sliced pieces off and extracted the RNA and figured out the genome sequence of the virus, synthesized the DNA copy, and put that in cells, and out popped 1918 influenza virus. So there was a recent TWiV with Jeff Taubenberger where he described this, and that's how we got it.
So not only, do you know how many independent isolates he did?
This is a good question. How many blocks from an individual? I don't know.
Not only that, but how many
How
different
many
individuals?
Yeah, I don't know. It's more than one, because then they also went up to Alaska and pulled a grave out, and that was a different person as well to complete the sequence. So I would say we don't know how closely this isolate mimics what was circulating, right?
Right, but we do know partly this is background in that we'll probably get to this, but in other animal models, this thing has different virulence properties than more recent viruses. So it supports the notion that you're dealing with a strain that has a special special virulence characteristics consistent with your preconceptions of what the 1918 flu ought to do.
Yeah, I think they said in the introduction it's highly virulent in mice and ferrets, right? And how that would compare to all the other pandemic strains, I don't think anyone's done them all at once, frankly.
Because other than this, we don't really know what was circulating then.
Right.
No,
Right.
we don't.
But just to show you how random nature is, there's no relationship genetically between a ferret and a mouse.
Are
No.
you sure?
Well, I mean, if you go back
Or a
to
human, for
vertebrates,
that matter.
they are both mammals.
Oh, yes, that's true. And they might, 20% homology?
Yeah,
30%?
they're very distant
They're
relatives,
as far apart
and they're
as you can
different
possibly imagine.
physiologies. You've got an herbivore and a carnivore. You've
There's
got
no
very
reason
different
for this
animals.
result except that's
Who's
what the result was.
Which one is the
Yeah.
carnivore, the ferret?
ferret.
Yeah, absolutely.
What do they eat? Mice?
They eat
Anything
get
they
their
can
little teeth on.
catch.
Anything they can get.
They are vicious little hunters.
They're weasels. They are
That's right.
vicious little hunters. They probably eat mice. They would certainly eat mice.
They do.
Hey, do you remember
yeah.
Weasels Ripped My Flesh? No?
Is that a movie?
Frank Zappa album.
Frank
okay.
Zappa album.
Of course.
Weasels Ripped My Flesh. We have some listeners who remember that. Okay, so in rhesus macaques, and then they also looked at replication in the lung. It replicates really poorly. Not surprising why it doesn't make them sick, right? Then they look at cynomolgus macaques, also non-lethal, and they put more virus in to these animals, but they don't get sick enough to warrant being euthanized, right? They put 7 times 10 to the 6 PFU. All the animals survive. They get more pronounced disease severity, but really still not. It's not lethal, and that's
You can almost sense the disappointment in this paper that, okay, here's this model. It ought to be a good model for pathogenesis of 1918 flu. We load these monkeys up with this virus. I mean, this would clearly kill most humans, I think. And
yeah, probably
and
would.
they're fine. They're little sick, little cough, but it doesn't seem to really affect them much at all.
I mean, they do a lot of work.
This is
here.
basically a negative results paper.
Yeah, they put more virus in, so you get a little more replication, you get a little more severity of symptoms, but they're not lethal, even with a high dose of virus. So, they do reproduce the virus a little bit, especially with the higher dose, but it's eventually cleared. And, in 3 weeks, 4 weeks after infection, there's no virus detectable. So they also measured 29 different chemokines and cytokines in the bronchoalveolar lavage, figure 6 with like 50 panels, so much work.
Yes.
Oh my gosh. And there's some, these are inflammatory cytokines, and they say maybe this is responsible for some of the pathology, but, and maybe
the monkeys weren't quite feeling their best.
No. So that's the story. And I think the take home message for me is that even despite a non-human primate, very close to humans, right? It does not recapitulate disease. And I think this is the problem with most animal models, is that you can never assume that they're completely recapitulating what's happening in people.
Mice lie and monkeys underestimate.
I have another question, though.
That's
I
right.
think a reasonable
Well, mice die in this case, but
a reasonable question that follows from this is that we had a series of tissues derived from the green monkey kidney cells.
Vervet monkey, yeah.
Vervet, and that means green.
It does?
It does, actually.
I'll look it up while you're talking.
Wondered why we hadn't tried this virus out in non-human primate tissue culture to see if it is infectious at all at any level.
Well, I'm sure it would be, but what's it going to tell you about lethality?
No,
animals?
it
I don't
won't,
think
but
it's gonna tell
it'll
you.
you may be able to identify a susceptible host that way. That's all.
And it doesn't mean green.
well, it clearly
Thought it
in
did.
this experiment we can see that it must replicate in non-human primate cells because they get
Yeah.
infectious virus out. There is a little bit of pathology. The monkeys are not perfectly fine. The virus is in there and it's replicating, but they don't get very sick.
Which monkey is the green monkey?
The vervet. We don't use green any longer. Apparently, there's some issue with using green So we use vervet monkeys. I don't know why, but Brienne
Oh,
does
okay.
when she's on the show. She
Right,
she
she
knows
would know.
why.
She would.
We don't call them green any longer.
Okay, fine.
So they're called vervet monkeys.
Right.
But it doesn't say in this article anything about green. Okay. Green is not present in this article. So it's been banished from the lexicon.
Green got canceled.
So non-human primates are not going to be useful for us looking at therapeutics. They have to find something else. Now they do. This is quite interesting in the discussion. They do say that it was a separate study of cynomolgus macaques in 1918 influenza virus infection, and they were some of them. 3 animals were sick enough to need euthanization on day 8, and they say these animals were older.
Uh-huh.
This was a Canadian colony that was depopulating the older macaques, and the animals ranged from 9 to 19 years of age. That makes me feel so bad. There's a 19-year-old.
I know. I had the same reaction
Yeah.
reading these details about the animals.
19-year-old animal who's lived 19 years, and you're going to sack it after this experiment.
Right.
It's
So-
just
Just briefly, could you describe the epidemiology of the pandemic of 1918 in terms of age groups of people?
So one of the
W.
devastating aspects of the 1918 flu was that it tended to kill young adults.
Correct.
Mm.
And not
Yeah.
the older people.
and not the older
No.
and the youngest, but in between, right?
Exactly.
And we don't quite know why.
So I know a rumor spread that when the American troops were asked to go home after the First World War, that a lot of them refused on that basis because they knew if they did, they would come home and catch flu. And a lot of them came home and caught flu, and they died.
That I have a hard time believing that anybody who was deployed to the trenches of
Alan,
World
I
War
agree with
I
you, and, you know,
was in
was a
any
horrible
way
war.
reluctant to go home.
But that's the point. The point is that this was such a horrible virus. They
Hmm.
were afraid of that even more than they were of
Okay,
the
maybe,
war that they
but
just got finished.
wow, that's. I've read about what was
You think those are
going
just
on
rumors?
in World War
Anyway,
One, and I can't imagine being afraid of the flu after
The
that.
reason I mentioned this Canadian colony with older macaques is because older macaques are immunosenescent, so that may account for the more severe disease.
Sure.
And that's one of the problems in using,
No, that's right.
different
Yeah.
ages. However, they do say that so these Mauritian macaques, which are used in this study, have low allelic diversity, so they're less diverse than other macaques. So that
Right.
may be why they're
Right. They point out that most macaques used in research are imported from. They're captured in a variety of mostly Southeast Asian countries, I think, and thrown together, and so you get a lot of genetic variation in most macaque studies. This one may have had a little less.
They do say that macaques, Mauritian macaques, cynomolgus macaques succumb uniformly to aerosolized H5N1 influenza virus.
Right.
So you could use it for that. So their conclusion is we shouldn't use rhesus macaques, but we could use 1918. If you give them a lot of virus, they get clinical signs that are similar to those of humans. They just don't die. It's not a lethal
Yeah.
model.
Right.
And I just want to remind you that most of the models for COVID are non-lethal except for the ACE2 transgenic mouse model, where it's lethal, and because there's a lot of receptor being made in those mice, and I think they die from a neurological disease, which is not related to why humans die of COVID, but it is used to as a lethal model, right?
Sure. Just to show you, I'm sorry to interrupt all this time, but
No,
I
it's
just
no
thought of
problem.
something. When I was at Rockefeller, Dr. William Traeger was a world expert on malaria, and he used for his primate, non-human primate, the Aeotis monkey.
Mm.
But the Aeotis monkey wasn't even susceptible to Plasmodium falciparum unless you splenectomized them. And once you did, not only were they susceptible, they all died. So the spleen plays a major role in some infections in terms of preventing
Yeah.
disease.
Yep.
And that's good to keep in mind at this juncture.
Aeotis, huh?
Aotis, A-O-T-E-S or A-O-T-U-S.
All
A
right.
smaller monkey.
So caveats of animal models. That's the lesson here. I hope we made it understandable.
Right.
Now,
Coke was wrong.
well, he didn't know everything.
No, but he thought he did. He did. He was a very arrogant individual, apparently.
Second paper is a little departure for us because it's about a cancer vaccine, really.
Right.
So we're not preventing a virus infection, but we're trying to prevent cancer or cure it even once it's established.
But this is cancer caused by a virus.
It is caused by a virus.
It's still in our territory.
Like hepatitis B.
It's within our purview, I guess. And it's also using mRNA vaccines as cancer vaccines, which we've mentioned a lot, but we never discussed. So I thought this would be cool. This is Science Translational Medicine, single immunizations of self-amplifying or non-replicating mRNA LNP vaccines control HPV-associated tumors in mice, where LNP is lipid nanoparticle and HPV is human papillomavirus.
Right.
Human papillomaviruses, or HPV, these are viruses, icosahedral capsids, nice protein shells with a double-stranded circular DNA genome, which cause warts of all kinds in many species, and they can also cause cancers. There are many, many different types of these viruses, and there are a handful that are associated with cancers in humans, cervical cancers, anogenital cancers, head and neck cancers. And in fact, what did they say here? That the fourth cervical cancer, the fourth most common type of cancer in women. Now, you may say, how do these viruses cause cancer? Well, it's very interesting. They have in their genome a protein, two proteins called E6 and E7. And E means early because they're made early in infection. The function of those proteins is to get whatever cell the virus enters to start dividing. Because a dividing cell is making DNA, and the virus doesn't have its own DNA replication system. It depends on the cell. So these two proteins, we call it, kick the cells into dividing. If the genes for those proteins should by accident be integrated into a host cell, the cell will keep dividing forever.
And that's called cancer.
And that puts it on the road to cancer, because every time a cell divides, it sustains mutations.
Right.
And if you get enough in the right genes, bingo, you get a cancer.
And certain HPV strains or serotypes are likely or much more likely to do this than others. So you know,
Right.
if you've ever had warts, they're quite likely benign and self-limiting, and everything's a cure for warts because they go away eventually. And
So, as a side,
but then there are a few serotypes of HPV that are likely to progress to, or more likely to progress to causing cancer.
Yes, Dixon.
as a side issue, I was led to believe, although you can obviously chime in here, that to develop a vaccine against human papillomavirus, there are a lot of cultures that are against that, because if you can prevent the ravages of HPV, this allows that person then to have sex.
Yeah, right. Okay. You're saying that having a vaccine against sexually transmitted
Right.
virus that causes cancer makes people more promiscuous.
That's their theory, not ours.
I just don't buy that.
No.
I mean, I was in a high school years ago and talking about this, and one of the kids said, why does my parent not want me to have this vaccine?
Exactly.
And I said, because they think you're going to be more promiscuous. And he laughed out loud.
No,
Yeah.
but that's true. That is true in some places.
It is
That
true.
people
in the more
believe that.
conservative places
I don't
in
know.
the world,
Yeah.
including the United States,
Well, that may be, yeah. Not the U.S., though.
you
There-
wouldn't think
yeah.
so. But
Oh yeah, there are
Ask
definitely
Richard.
people
Rich,
who
don't you
believe
think
that
they're-
and who oppose this vaccine for that reason. But if you think about the subtext of this, they're essentially saying, I would rather my kid get cancer than
Right.
have sex outside of marriage.
that is exactly right.
Which is, I'm
Insane.
sorry,
It's
insane.
insane.
Well, but humanity
right.
is full of insanity, right?
So anyway, yeah, there is an effective HPV vaccine. It is now part of the standard immunization series. It's given to adolescents. and should be given to all adolescents and up to, I think, if you didn't get it, you can get it up to age like 20 something, and it's still quite likely to be
effective. Go up to
effective.
45 now. And
So men can catch this
they
too.
boosted the age more and more.
men and women can both get it. They both need to be vaccinated. And by the way, this paper is from University of São Paulo, University of Pennsylvania, and BioNTech. You know that company?
Sounds familiar.
In Germany.
Is it
That's the
BioNTech
mRNA
or
company,
BioNTech?
BioNTech, sorry, BioNTech,
BioNTech,
yes. I'm sorry.
That's right.
A made-up name I pronounced incorrectly, yeah.
Yes.
And the first author is Jamile Ramos da Silva, and the last author is Luis Carlos de Souza Ferreira. And we have here Catalin Carrico in this list because she's working with or for BioNTech. So you can, as Alan said, you could vaccinate against the virus and prevent the development of cancer. But what if you already have an HPV cancer?
What about us old folks who missed the window of opportunity to get this
Could you-
vaccine because it didn't exist?
Could you make a vaccine that would get rid of the cancer? That's what this paper is about. And they're all excited about mRNA vaccines, right? So they're going to apply the approach for cancer, but they have some twists which are really interesting. So we have, the mRNA vaccines that were used for COVID, their RNAs are modified bases, so they don't activate the immune system as well, and they make more protein. So modified RNA, non-replicating. Then they also compare that to a non-modified RNA, which is going to trigger an immune response more so. Maybe for a cancer, that's good, right?
Sure.
So we're going to and then we're going to look at self-replicating vaccines, which is very interesting. In these cases, these are derived from viruses. So you have basically, there are several ways to do this, but in the simplest way, you can make an RNA having a coding region for the viral protein, the RNA polymerase that copies the RNA or makes mRNA. And then you have a gene, in this case, the E7 protein that we talked about. And this is self-amplifying because the polymerase is made from that mRNA, and then it makes mRNA from it to make the protein. You can put DNA in as well. You can put virus in. But in this paper, they're putting RNA. They're injecting RNA into their animal model, which happens to be a mouse. So they're comparing them, all these
and
different...
in addition to the HPV E7 protein, they're also including the sequences for herpes simplex virus glycoprotein D.
Yeah.
Which they say, and actually this is it's paywalled, so probably most people aren't even going to be able to read this paper. But they mentioned this in the introduction, and I was then going through the whole rest of the paper asking, why did you put glycoprotein D? Am I supposed to know this? And they finally mention in one line in the discussion why they did this. And I had already looked up the reference that they gave to an earlier paper. This protein has like an adjuvant effect, I gather. It boosts the immune response. And so they've included not only the target protein, the E7 protein, but also this glycoprotein D that's going to kind of boost the immune response toward the direction that they want it to go.
It's actually a fusion, isn't it? Is it
yes.
not? Between
So they fused
glycoprotein
these
D and
two.
E7 protein.
The mRNA encodes a fused protein that's glycoprotein D and E7.
So I'm
And just to emphasize on the self-replicating RNA, the idea there is that since it's self-replicating, you get more copies of the mRNA and theoretically more protein, theoretically a more robust immune response.
Right.
But because it's self-replicating, it's also going to be unmodified, right?
Right. It's unmodified. That's right.
So let's go back to the virus itself.
Yep.
When the virus infects a cell, and of course, you already said what happens, they secrete a couple of proteins, which deregulates cell division.
Right.
And it makes more cells available to the viruses after they kill the cell that they just went into. But many viruses don't.
I don't think that's correct.
Well, what selective advantage does this have over a virus that goes into a cell and doesn't induce mitosis.
Well, yeah, if you're a
altering.
DNA virus and you do not encode a DNA replication system in your genome, and these are small genomes, so they have no DNA polymerase, you have to get the cell to divide.
I got you.
But say herpes viruses and pox viruses, which have all the DNA synthesis machinery, they don't care. As Rich would say, they avoid the nuclear bureaucracy.
Yeah, because cells in their resting state, in the like most of the cells in your body, the somatic cells, they're not replicating. Their DNA polymerases are just sitting around.
Like many of these other small DNA containing viruses, the only thing that this encodes relevant to DNA replication, aside from these stimulatory proteins, is a protein that binds to the viral DNA and directs all of the other host machinery to interact with that genome.
Gotcha.
You know, waving a big flag, here.
And
replicate this guy.
Okay,
and
that makes
and
sense.
replicate that genome.
Rich is studiously avoiding the jargon word, which the
There are
origin
many that just passed up.
the origin of replication, which is where the polymerase binds and this protein binds as well. Yeah. So that's why, Dixon, you need to get the cell to divide. And that's why I say it doesn't help the neighboring cell because the E7 protein has to be made in the cell where the virus is.
Okay.
Yeah. It doesn't move from one cell.
And it
to
doesn't
another.
kill the cell it's in?
Well, do these papillomas kill cells? They don't.
Well, so what happens is that in the situation with the warts is that through a break in the skin, the virus is introduced into the basal cell of the epidermis, which is where all of the cells that make up the epidermis originate. And it infects that cell and goes through part of the replication process, making early proteins. And then those cells, in their normal differentiation process, migrate up through the epithelium and differentiate as they go until they form essentially dead bags of keratin up on the top, keratinocytes, okay? As they migrate up, this is fascinating. The virus transcription program responds to the environmental signals of the epidermis, the differentiation signals in the epidermis, to actually change the program of virus replication so that once you get a ways up in the epidermis, and the cell's already looking a little funky. I mean, it's got a cytopathic effect that's recognizable, which is part of the way they diagnose a cervical carcinoma. Then you trigger the late stages of virus replication. And in the upper levels of the epidermis, you're actually making virus, and those cells eventually get up to become keratinocytes and are shed. Now, I don't know if their fate is death, even if they're not infected.
Right.
Okay?
Yeah.
Whether or not the virus actually manages to kill them before they would die, I don't know the answer to that. But it doesn't really matter because they wind up sloughing off in the epidermis. But your question is important because, especially in the early parts, you get these proteins that stimulate the cell DNA replication apparatus actually do stimulate the cells to divide. So you get a hyperproliferation of the cells that are in these early programs of DNA replication. And that's why you get a wart. A wart's a tumor.
Right.
Okay?
In addition, to get a tumor, the virus cannot kill the cells, obviously, right? Because the tumors are living forever. And so in those cases, it's just the E6 or E7 protein that's in the cell.
Right.
It's accidentally come away from the genome, integrated.
I hear you.
And in fact, if you remember Henrietta Lacks,
Sure.
so she had a cervical tumor. That's what killed her.
All right.
They took a piece of it out and made the famous HeLa cells, right, that grow forever because they're immortal. Many years later, they sequenced that viral genome, and what do you think they found integrated into one of the chromosomes?
E6.
E6.
E6 or E7, I don't remember, of
E6
type
or E, yeah.
18. Type 18 papillomavirus.
Wow.
Showing you exactly why she got a cervical tumor. It's just amazing.
Wow, indeed.
All right, so this is a very methodical and meticulous paper. First, they ask, if we simply put these RNAs into cells, do we see protein? Do we see this fusion of E7 and GD? And how much do we see? So we have methylpseudouridine modified or unmodified mRNAs. Remember, the COVID vaccines that you got, they had methyl pseudouridine containing RNA. So it's a modified U.
It's a chemically modified RNA that is considered better for vaccines.
Works better
for
for viral vaccine. Then we have unmodified, and then we have self-amplifying mRNAs. They put them into cells, and they say, what kind of proteins do we get? The U self-amplifying outperforms everybody by tenfold. Hey, it's a self-amplifying, right?
Yeah.
Somebody blowing leaves outside?
Yeah, sorry, I'll
No,
mute
it's
that.
okay. No worries.
I could close the window, but my cat would be disappointed.
That's all right. We'll just remove it in post.
Oh, wow.
They're harvesting lupin.
It's okay. Then the non-replicating similar levels of proteins. Okay, this is in cell culture. And they also looked in mice. They injected them intramuscularly so they encapsulate these RNAs in lipid nanoparticles to protect them, and then the cells will take them up more readily. And then they measure, in this case, they have a gene that encodes a protein that glows and makes light. So they can easily measure it in the mice, and they do whole body.
And that is green, by the way.
Well, it's not green fluorescent.
It's not green in this
It's a
case?
luciferase.
This is
Oh, okay,
luciferase.
luciferase.
It's
But
a
then
yellow
they can put the
fluorescence.
mice in a
Jello.
machine that can see the luciferase in the whole body. It's just remarkable. And what is it? There's some differences here. The self-amplifying, unmodified, they see luciferase expression for 23 days. The modified RNA had similar kinetics, but they declined more rapidly.
Yeah, it's not huge differences in the grand scheme of things.
No.
These all get expressed and they all stick
But
around.
they're expressed. That's the point.
For varying lengths of
You're
time.
making protein, which means now you can go and do some biological experiments. So first they immunize. So they want to say, well, what are the kinds of responses we get? So they're going to immunize mice with these various RNA formulations. So they inject, and I have to say here, now they have a cell culture. They're called TC1 cells. They're going to inject into mice, and they're going to grow into a tumor. And they say these are accepted as a model for HPV-induced tumor formation. So TC1 cells, they inject them on day 0, and then they immunize the mice with RNA 3 days later.
So these TC1 cells are an HPV-driven
Right.
tumor cell type, right?
Exactly right. So they're making
E7.
and E7, they're grown forever, and you inject them in
Yeah.
mice, they make tumors, yeah.
And if you don't treat these mice with anything, this tumor
It grows
gonna
and grows, and you have to
grow and
have
grow.
to sacrifice them because you can't let them have huge tumors. It's not.
Yeah.
So the idea is that E7 is a marker for this tumor and essential for the tumor to grow. So if you could have an immune response to that protein,
Exactly.
you might be able to wipe out these cells.
Exactly
Yes.
right. Yeah. Okay, so the first thing they do is measure CD8 positive T cells. So these are a kind of T cell that will kill a tumor cell, basically. And so they're assumed to be important for clearing a tumor, as opposed to antibodies, right, which are great for viruses, for blocking infection of cells, but probably not going to do much for a tumor. It's the cytotoxic T lymphocytes that are going to do it. And when they immunize mice with these formulations, they see increases in cytotoxic T lymphocytes that are specific for the E7 protein, which is what you would like to do. They all do this, some higher than others. They show that these cytotoxic T cells are functional. They will kill cells in culture that are producing the E7 protein. So they're not just there, but they're functional. They're also polyfunctional. They not only kill cells, but they make multiple, 2 or 3 different cytokines. As Brienne would say, they have multiple activities, which is what you would like to see. And so all of these RNAs can induce these polyfunctional T cells. Okay. The other question they asked, which is very interesting, is if if the mice actually have these TC1 cells that are making E7, does that actually help, the vaccine? Because you're giving them E7 basically to try and make a T cell response. So does having E7 in the mouse help? And the answer is yes. If you compare mice with and without the tumors, you get higher numbers of T cells when the mice have E7 cells into them. And then what about the tumor? Does the tumor get any smaller? So now remember, we're injecting mice with these cells, and then we're giving them the vaccine. So in this experiment, they put the TC1 cells in, and then they give them either some control mRNA or one of these specific mRNAs. The control mice, the tumor keeps growing until they have to euthanize them. But in mice immunized with these various RNAs, the modified, non-modified, non-replicating, or replicating, 100% survival, complete protection against tumor growth. And all mice are tumor-free at the end of the period, 7 days. So in a mouse, you'd stick in these cells, they would get huge tumors, and they would have to be sacrificed. If you give them these mRNAs, you have no tumors left. They don't ever grow. Isn't that amazing?
They mount a robust immune response against the tumors and take them out.
Yeah.
Yeah.
Also, they wanted to know if they could give lower doses. So they do an experiment where they engraft mice with cells, they immunize them, and then they challenge them again with a second dose of tumor cells, but without giving them another dose of vaccine. And they have not 100% protection, but 5 micrograms can do 50 to 80% protection on a second dose. The first dose was completely protected. The second dose is not bad. And what is mediating this protection? It turns out to be our cytotoxic T cells. They do a series of experiments where they show, in the mice where they're protected from tumor development, they have high levels of cytotoxic T cells. They also have memory T cells, which is what you would like to see made. And it's a correlation, right? So high levels of these T cells correlate with tumor protection, but they actually do an experiment to prove it. They get mice lacking either CD8 or CD4 cells. So the CD8s are the cytotoxic T cells. The CD4s produce cytokines that help the CD8s become functional. So they have mice lacking either kind of cell. If you take the mice lacking CD8 cells and you put the tumor cells in them, you give them the vaccine, no protection against tumor formation if you don't have CD8 cells. It's really tells you that the CD8s are really important.
Yeah. It's a great experiment because, we give this vaccine and the tumor goes away and the second tumor we implant gets taken out, and that's all great, but does it work the way we think it works is always a really important question. And the answer here is it seems like it's really working through the CD8, the killer T cells. These are what's taking it out.
And so this is not an experiment you can do in people.
No.
So this is an example of where you have a mouse model which you can genetically manipulate, and it will tell you, okay, CD8 cells are important. So then you could look in people, if you're going to do a trial with this vaccine, and see if it induces CD8 cells, if that's protective, and so forth. The CD4 knockouts get partial protection against tumors because, as I said, the CD4 cells are needed for development of CD8 cells. So it's kind of an indirect effect. So these experiments are all done by putting the tumor cells in and a couple days later putting the vaccine in. But, in the real world, it's not how it's going to work. You're going to have a tumor already.
You don't diagnose the tumor within days of somebody having started to develop
Dixon?
it.
Right.
Yeah.
So now the question is, if you have a tumor, can we inject some RNA in you and will it go away?
Exactly.
Established can it eradicate established tumors? So they put the TC1 cells in mice. When they get 12 millimeters square in size, then they're given the vaccine. So if you put the control vaccine RNA in the mice, the tumors keep growing. They have to sacrifice the mice. But mice given one dose, 5 micrograms of any of these 3 RNA vaccines, eradication of tumors in 60% of the animals, survival for 100 days. So,
and 100 days is quite a while in a mouse lifespan.
I suppose you could tweak this and get better survival.
So one of the things that occurs to me looking at the data from this is that in the in the higher dose in particular, it's not as clear in the lower dose, but in the higher dose, when you do this experiment, it looks like, measuring tumor area over time, that all of these animals, the tumor continues to grow for a little while after the immunization. And then in all the animals, the tumor decreases in size. And then in some of the animals, the tumor comes back. Mm-hmm. And I have to wonder, are those tumors different? Is there, are they somehow evading this thing or not? So I would assume that that would be a subject for study down the road.
Yeah, it could be that they're different, or it could be that some were left in complete eradication, and then the CD8 levels went down, and maybe they grew back. But yeah,
maybe this is a situation. I mean, they make a big deal about how this is a one-dose thing.
Yeah.
Yes.
Maybe you need to boost them.
Yeah, it wouldn't be bad to have to give a couple. People would love to have their tumors taken out by a couple of shots, right? Most people.
Yeah, and this is a pretty extreme test compared to the way cancer is generally treated in humans, where you don't give one shot of a monotherapy.
Yeah.
You know, you'd go in, and if it can be surgically removed, it'll be surgically removed. Maybe there's radiotherapy. Maybe you get this. Maybe you get, you know. So these are typically cancers that are treated on multiple fronts.
Then they also do an experiment where it says, okay, these, we've got tumors all over these mice, but in HPV tumors, they're in certain places. So let's see if we put in cervical or head and neck tumors into these animals, can we eradicate them with these vaccines? So they have intravaginal and sublingual orthotopic. So they put the tumor cells intravaginally or sublingually.
Or under the tongue.
Or under the tongue. You get tumors in those places, which is where HPV tumors are seen. Then they do therapy. They immunize the mice with these vaccines 3 days later. And complete tumor regression in the intravaginal tumor model with the non-replicating or the self-amplifying RNA. All mice tumor-free 2 weeks later. And then protection by the, this is interesting, the pseudouridine modified RNA is slightly lower protection. So it may be that the features, the pseudouridine, which is good for a viral vaccine, is not good for inducing T cells that are going to kill a tumor. So that was the intravaginal model. They do the sublingual model, complete tumor regression in all treated mice with the non-replicating or the U-replicating. That means unmodified RNAs. And partially protected again with the pseudouridine modified RNA. So it's not as good. And finally, they compare these 3 RNA vaccines to previously tested DNA and protein-based GDE7 fusion vaccines. So others have tried, or maybe they did, I don't remember, DNA-based vaccines where you encode the fusion protein into DNA, you inject the DNA in, or you make the protein and you inject the protein, kind of a Novavax approach, right?
Yeah.
Same modality that we've been talking about. the
And
RNA
this is an
vaccines are
important-
clearly better, right?
Yeah, this is an important comparison because people have worked on these vaccines, and my understanding is that the results have not been all that great.
Yeah.
So if those had worked really well in this system, then you would say, oh, well, just everything works in this system. But to see this result where something that hasn't worked so well doesn't work so well here either, but the mRNA really nails it.
Yeah, the mRNA. So this is a they inject the cells and they vaccinate. The RNA vaccines, they do great at eliminating the tumors. 80% of the mice tumor regression, survival up to 90 days. No animals that received a single dose of the plasma DNA or the protein survived tumor challenge, similar to luciferase control. Not very good.
Yeah, and in this one, they did something of a hybrid of their previous approaches. So they give the TC1 cells, they let the tumor get established,
Right.
they let it get to 9 square millimeters in this case, so not quite as big as before, but I think it's 10 days later. Yeah, 10 days later. So they let some time elapse and then gave the vaccine. So it's
So the
kind of
mRNA
realistic.
is only against a single protein?
It's
or
a
is there
fusion
multiple- Three
of-
different
the fusion protein.
No, two proteins. A fusion
sorry.
of the E7 with a herpes virus protein that seems to be an immunostimulatory
Yeah, that-
protein.
And what's the other one?
No, E7 is the oncoprotein.
Oh, okay.
And then there's 3 different RNA formulations,
Okay.
right? There's a modified mRNA, there's an unmodified mRNA,
right,
then there's a
sure.
self-replicating
Sure.
mRNA. And I think the unmodifieds are better,
Yeah,
for
they
this.
seem to be better in
right?
here.
So I think this is very, very exciting findings, that these can clear
Yeah,
established tumors. They say mucosal tumors are hard to do immunotherapy because you can't get cytotoxic T cells in there. And so these are doing a great job at doing that. And they also say that this tumor cell mouse model is accepted as a preclinical efficacy data and can be used for the proposal of clinical evaluation. So in other words, you can take data from these cells injected into mice and go to the FDA and say, we want to do a clinical trial. Now, you don't need to have a non-human primate, for example.
right.
They also mentioned, so this E7 is from HPV 16, the epidemiologically relevant strain, but they're also 18, 31, 33, and 45 are associated with cervical cancer, so they should test those too, right?
Yeah.
I wonder if they would make a multivalent vaccine.
Well, the HPV vaccine that's now part of the standard immunization regimen is multivalent,
Mm-hmm.
and I think it hits all of the ones that are known to be major cancer causes.
Multivalent means multiple proteins.
Multiple proteins. So it's
proteins,
targeting
yeah.
multiple serotypes of the virus that could
So
cause cancer.
I suppose it would be advantageous to make a multivalent cancer vaccine, right?
Yeah.
Instead of having to type your cancer and then decide which one to use. But it could be that combining, 1, 2, 3, 4, 5, it reduces the amount of each mRNA, and maybe
It may.
they'd have to do these experiments in mice to figure that out.
It's not clear to me how much cross-reactivity amongst the E7 proteins, how much homology amongst the E7 proteins there might be, and how necessary that might be. Certainly when you're talking about different, effectively different serotypes, where you know that there's an immunological difference between the capsids for the capsid protein vaccines, you're going to need a multivalent vaccine. It could be that there's sufficient homology among the E7 proteins so that you don't need as many, or maybe you only need one. But this is just my ignorance. I don't know how much homology there is.
It may be that the T cell epitopes, which are the ones that are important, are more conserved among the different serotypes, right? Because the serotypes are defined on antibody, I guess, right? So,
yeah, and the vaccine, right. So the preventive vaccine needs to induce immunity against the capsid or surface proteins, and those differ.
right.
And this E7-based vaccine, you're aiming at an internal protein that probably differs less because it's a conserved function.
Actually, this is a little bit of semi-trivia here, but you made the statement that the serology is based on the capsids. In fact, the HPVs, since they're you don't get a good antibody response to papillomaviruses. So they not only that, but you can't grow them in culture. The typical the way you do serotyping is to take serum from somebody and react it with the virus and see if it neutralizes the virus in culture. Well, in this particular case, you don't have any serum because you don't have any antibodies in the serum, and you can't grow the virus, so you can't do that. So what they did with the HPVs was they simply sequenced all the DNAs, and they made a more or less arbitrary decision that any two viruses that were different by more than 50% were called a different type.
Okay.
But it turns out that that was accurate because a cancer vaccine against one type is not necessarily cross-reactive against another.
Okay.
So it worked.
Okay. So when I said that, I said, I have a feeling this is wrong. So I'm glad you corrected it.
Thank you. Yes.
It wasn't wrong. This is just an elaboration.
So
okay?
the other point is that when they compare the genome sequences, were they looking at just a capsid protein, right? That is one.
I think it was the whole genome.
The whole genome. Okay. I just wonder if the T cell epitopes are just more conserved anyway.
Sure.
The vaccine, the preventive vaccine, is an empty capsid, right? So it is
correct.
Making antibodies and T cells against those epitopes.
Right. So we're talking about a completely different viral protein than in these guys, with a completely different function, with respect, even immunologically. So it could be a different story. It just remains to be seen.
Anyway, so this is against cancer, and the existing HPV vaccines are to prevent infection in the first place. So we need this in addition.
Yeah.
Because many people don't get immunized and they get cancers, so we need to be able to treat them. So I don't know. It sounds like they want to go into clinical trials now, right?
Yeah.
Go for it.
Yeah, go for it.
I think it's a great idea.
Do it. Let's do a couple of emails. Dixon, did you find any jargon in that discussion of?
Only minimal.
I think we all made a conscious effort.
I hope, in fact, Jeff writes us back and tells us how we did.
Yes.
Yeah, Jeff, how did we do?
Let us know.
Did you get it? Okay, Rich, can you take the first one?
Sure. Paul writes, Dr. Racaniello, I was infected with COVID before vaccines became available, as were many others. We didn't have a choice to be vaccinated. To my surprise, the public health apparatus immediately cast doubt on immunity from infection, which was fine initially, but within a year there was substantial research looking at the immune response from infection. All the research pointed to a robust response. Upon vaccines being introduced, the same type of research was conducted and found a similar robust response. To this day, public health has refused to admit the high quality of immunity received from natural infection, and I would argue better immunity in numerous ways. Indeed, no one should risk infection without vaccination. Vaccination should be considered a bridge to more safely be infected. That said, for public health to deny infection-derived immunity, whether actively or passively, is scientifically proven, creates mistrust. People want the truth. They despise when people in authority withhold information in order to achieve a desired outcome. I know better than you. Public health should fully admit the supporting immunological data on infection and also recommend strongly to be vaccinated for safety. The work of scientists like Shane Crotty, Michelle Nussensweig, Florian Kramer, Jackson Turner, and many others has shown that in almost every capacity, infection bests vaccine in establishing underlying immunity other than a lower, more heterogeneous initial antibody response. greater memory B cell magnitude and diversity, greater memory T cell magnitude and diversity, lower antibody decay rate, substantially greater dimeric IgA response in the upper mucosa, substantially greater tissue resident memory in the upper mucosa. Why can't public health share these facts? Are ordinary people incapable of making their own decisions? I would also argue that public health's charade on infection has caused harm. Recommending vaccines at 3 months post-infection is maddening. Some somatic hypermutation after infection can last for up to a year. As you know, this phenomenal process can greatly broaden one's memory B cell repertoire. Vaccinating at 3 months stalls, if not disrupts altogether, this process. Why on earth would public health rob someone of this benefit after having gone through an infection, especially knowing that infection typically prevents reinfection for 6 months or more? As always, love the show.
Good points.
Good points.
Well, kind of. But I have to say, I've never met public health. So I don't know why he's going around saying all these things that I haven't necessarily heard coming from public health agencies per se. I have not seen anything from the CDC that says infection doesn't provide immunity. So I think there's a bit of a straw man going on here, or just maybe a misperception or a different perception.
Well, I don't know if you've seen just in the last few weeks, the Lancet article came out about how good natural infection is, right? And the coverage of that article has been the Lancet finally acknowledges natural infection, right? Mm-hmm. So I think it's a combination of the journals. I think it's a combination of the commentaries, the pundits who are talking about this, were saying, natural infection isn't good enough. It's not correct, right?
No, I don't think that argument has really ever been like a major thing that public health experts or authorities are making. People have acknowledged from the beginning, if you get the virus, you're going to develop some immunity. It doesn't protect you from subsequent infection, but neither does the vaccine. But it's providing some degree of protection against serious disease, sure. I think that's known. It's not something that has been hyped because of the risk that people would then say, oh, I don't need to get the vaccine, I'll just get the infection. I'm young and healthy, and so it's not going to be a big deal. The other thing to bear in mind about this recommendation of the 3-month vaccination is the people at highest risk of dying from COVID-19 are folks with senescent immune systems, who may very well benefit from a boost at 3 months.
Sure.
Whereas somebody whose immune system is younger might not necessarily benefit from that, but you make the recommendation on the basis of the people who are most likely to die.
But don't you remember early on when you had to be vaccinated to do certain things? A natural infection didn't count.
Yes.
I think that's part of Paul's recollection of this narrative. Yeah.
Okay. I'm not sure that was really presented as an argument that natural immunity was not acceptable. I think it was just we can verify the vaccination and we know the level of immunity that gives.
Maybe.
Whereas natural infection is more of a crapshoot.
Yeah.
And also, I'm sure there was at least some discussion of the fact that if you accept natural infection as as equivalent to vaccination, then that maybe encourage people to go out and just get infected rather than get vaccinated, which was a very dangerous thing to do. So yeah, I agree, Paul, that natural infection provides immunity, and I think if people are going around saying that it doesn't, then they should be corrected, but I'm not seeing a lot of that argument being made, except as a straw man.
I had the same kind of reaction to the letter, Alan, and I have nothing to add. I think that's a really good analysis of the whole situation.
Alan, can you take the next one?
Okay, this is a follow-up. Paul writes, as a follow-up to my previous email, I've attached this paper by Shane Crotty, which you may have already read. It's one of the most complete papers I've seen covering the totality of COVID immunology and is supportive of the email I sent before, provides a link. One aspect that is missing is the immune response post-second infection without vaccine. Although there may be more heterogeneity than with hybrid immunity, I suspect that the quality of the response is much like hybrid. It seems the preponderance of quality that comes from hybrid immunity is a product of the engagement via antigen challenge of the quiescent immune memory created by infection. As for vaccination recommendation by CDC at 3 months, I waited 10 months for 1 dose of Pfizer, not 2. I based my decision on understanding somatic hypermutation and affinity maturation and 15 research papers that determined the second dose of a prime series did not offer any additional benefit. Florian Kramer, whom you've had on TWiV, was an author on one of these papers. I was taught how to do research, how to do it without bias, to always present all the facts, to make recommendations, and let the recipient make their own choices. Thanks, Paul. I mean, yeah, if you want to second guess the recommendations, you're certainly free to do that. That is a way to approach it.
I don't think everyone can do that, though. That's why most people have to follow public health recommendations, right?
Or should. And I wouldn't recommend most people taking matters into their own hands like this because there's a very, very high risk of motivated reasoning taking place, especially if you're doing this in isolation and saying you did your research, okay, but how much do you really know about how the immune response is going to play out? Whereas if you stick with the official recommendation, you at least know that if that wasn't the right move, you're going to be in a situation that others are also in, and this can perhaps be addressed.
Dixon.
Vincent. Gabriella writes, "Hi, Twivers. Love the show. I began listening when I started learning English when I was in college in Mexico in 2009 or 2010. As a veterinarian that later became a virologist, I must say that you have a huge influence on my career. You have had a huge influence on my career path. Thank you for all the science communication you have given to the world. On TWiV 987, Arturo mentions convalescent plasma, while in your legend, it is called convalescent serum. Serum and plasma both come from the liquid portion of blood that remains once the cells are removed, as Arturo described. But that's where the similarities end. Serum is the liquid that remains after the blood is clotted. Plasma is the liquid that remains when clotting is prevented with the addition of an anticoagulant, for example, EDTA. Therefore, the plasma contains other things like fibrinogen. I heard Twivers interchangeably use these two words, but they are not. Hope this helps to differentiate them. Love y'all, Gabby Toomer."
Yeah, I looked back in the in the show notes for that episode. I wrote serum. I don't know why. I went back and corrected it all. And yeah, I think we just often fall into using them incorrectly. But it's plasma.
I think it's because we're frequently thinking of anti-sera.
Yeah,
That's
maybe,
right.
maybe.
That's right.
And anti-serum meaning antibodies, and then serum is just a natural extension. At least that, with me, that happens automatically. But yes, these are two different substances.
No question.
As for me, as we were recording that episode, I was recalling that there is a difference, but I couldn't remember which was what or exactly what the difference was. Okay, and so I'm glad to have this clear it up. Now, with respect to what is actually used, Arturo said they make this product by plasmapheresis, right? Which is basically filtering your blood through a machine that gives you back your cells and takes out the plasma. And I assume, since it's called plasma therapy, that they are using plasma, not serum.
Yes.
Correct. Thank you, Gabby. And Desiree writes, I have heard you mention on your show that the mRNA COVID vaccines are essentially a 3-dose vaccine. Is this true for everyone? I have a healthy 9 and 12-year-old boys that received 2 doses in December. I am a healthy 42-year-old woman with no underlying conditions. I received 2 doses in July of 2021. Is it essential that myself and 2 children receive the 3rd dose? We have not had COVID. Up until January of this year, we have been continuing to be extremely safe, masking kids attending virtual school due to my mother having a bone marrow transplant, living with us, and needing to stay as healthy as possible. Now the kids are back in school. We are getting back out into the world. I am wondering if we are still protected at all. I cannot seem to find any studies regarding the longevity of T cells after mRNA vaccines. I am not interested in vaccinating to increase antibodies and prevent infection for a few months, but I do want to be sure we are still protected from the worst case scenarios. Hey, Desiree, that's great. You're absolutely right. That's what Paul Offis said. Let's not boost, except in certain populations. And it doesn't sound that you fit the populations. However,
Well, wait, Paul was talking about the 4th shot, though, wasn't he?
Yeah.
Yeah.
yeah, but she mentions boosting for preventing.
Oh,
you know.
right. So,
Yeah,
so
you're
this
right.
question, I
4th
read
shot.
this question as, should we get a 3rd shot? And my answer would
Yes.
be yes.
Yes.
Agreed.
Because we do have, and I think we've talked about these on TWiV. There are a number of papers that show that the 3rd dose gives you a broadening of the response compared to the 2-dose regimen, and that part of it is definitely worthwhile.
Yes.
The question is the 4th dose, which
Right.
we've all kind of been. I mean, the data are not real strong, and Paul Offit made a good point that it's not necessarily accomplishing much, and it probably doesn't need to be tailored to whatever the variant du jour is. I got my 4th dose, and probably, I'm sure I really didn't need to. My wife didn't get her 4th dose and isn't required to and isn't going to. If people want to get a 4th dose, fine. I'm not going to encourage it.
Let's do another round. Rich.
But third dose definitely yes.
Yeah, 3rd
Yeah,
for sure
I would
because
say
of the
the same
broadening.
thing.
Yeah.
Matter of fact, that 3rd dose is fascinating. Between the
Yes.
2nd dose and the 3rd dose is when the somatic hypermutation and maturation of the immune response goes on. And that 3rd dose is, to me, just is fascinating. And it does give you a really much broader immunity. So Grace writes, my name is Grace, and I'm a 2nd-year student at Caltech studying biology. I have a few questions in regards to Paxlovid. I am very curious as to what makes the drug specific to targeting enzymes that helps COVID viruses replicate. How does Paxlovid not interfere with enzymes in healthy cells? All right, she's right on it. Also, is it possible that Paxlovid or other drugs similar to it can be used to lessen the impacts of other viruses besides COVID? Yes! Great! Actually, I don't have a specific answer to this question, except that it's a perfect question.
Exactly the right questions.
I think you, these are all the issues with antivirals. How do you make a drug that's specific to the virus when the virus is using the cellular machinery? Okay, you got to have a drug that's somehow specific for the virus, and once you've done that, will it work on other viruses as well? And I think my answer to this would be, although I can't quote you chapter and verse, that I believe that the Paxlovid targets a viral protease, okay, and that there are sufficient differences between the viral protease as being specific for that virus infection and similar cellular proteases so that you can come up with a drug that'll target that particular viral protease. Not only that, but there are probably differences between that and other viral proteases as well. So proteases are becoming really pretty good targets for antivirals because of these very facts. There are specificities. Many, if not most, viruses use proteases of some sort. That is, these are enzymes that cleave proteins for various reasons during the course of a virus infection. I think there's enough specificity in exactly what those enzymes are doing so that you can design drugs that are discriminate between the virus and the cell or the virus and other viruses. The cool thing would be to come up with one that did a bunch of different viruses and none of the cell targets. But that's asking a lot. Any pushback? Is that did I do
good.
okay?
I think you nailed it.
We did the paper from Pfizer where they identified Paxlovid and showed that it actually inhibits other related coronaviruses.
Okay, that makes sense.
So, I can't remember which TWiV it was, but it's very interesting because, you have an active site of the protease, and you can make sure that whatever compounds you design are specific for that. You just do screens, and you show that they don't have toxicity to cells, for example, which would suggest that they're not inhibiting anything in a cell. Right. Alan, can you take the next one?
And we're up to
Beth.
Beth. Hello all. In light of the discussion of the DOE report on the origin of SARS-CoV-2, I think you will appreciate a couple of pieces I read recently. In Intelligence Analysis is Not Scientific Investigation, Cheryl Rofer explains why intelligence analysis is not a good way to investigate the origins of the pandemic. The other piece by Jonathan Katz is in the March 1st edition of his Substack newsletter, The Racket. It's called There is no Lab Leak Theory, with the subhead, It's Still Just Vibes in Search of a Hypothesis. His thesis is right there. People claiming the pandemic is the result of a leak of the virus from a lab don't even have a theory to explain it. They have a bunch of assertions. Both articles focus on the fact that the lab leak proponents have no data to back up their supposition. Yeah.
I actually went and read both these articles, and
Okay.
I would recommend the Jonathan Katz article in particular. The first one is just sort of a short note. The Jonathan Katz article is actually quite well written, and this is an individual who has some expertise in this. He was the guy who tracked down the fact that a cholera epidemic in Haiti was, in fact, of human origin.
Right.
Workers, I guess, with the UN or the
UN.
With the
Yeah.
UN, who had gone in to help out. I don't know the details, but managed to contaminate the water with cholera and precipitate that epidemic. So Katz knows what he's talking about, and he knows how to do this kind of thing. And he does a nice comparison between the cholera epidemic investigation and the lab leak hypothesis, and said, I love this phrase, there's no there.
Yeah. Yep. No data. It's absolutely bad. But if you tell that to them, they get very mad at you.
It's okay.
Yes.
Get mad. Fine.
Dixon.
Ken writes, Vincent, I'm a pleased patron supporter of TWiV and found an unexpected bonus amongst all the enlightening discussions in a recent TWiV podcast. Your recommendation for the application of Grammarly has convinced me to add that extremely useful computer application to my computer. I am so impressed with the free version that I will follow your lead and purchase the premium version to show my support. In preparing to invest in the upgrade, I came across the following information on their webpage that will enable me to make a very meaningful donation to the people of Ukraine. And she lists the web address. Might I suggest that a mention of the relationship between the people at Grammarly and Ukraine on TWiV would undoubtedly help in their Stand with Ukraine effort. Best wishes and thank you and the other TWivers for providing a pond of rationality and knowledge in a vast ocean of inane and ignorant humans. So well put.
Well, I didn't realize this. Grammarly was founded in Ukraine.
No idea.
Our co-founders are from Ukraine, and we have many team members who call Ukraine home.
Oh, wow.
And they say, we please to stand with you completely and wholeheartedly, and we are sending you aid. And they have, Grammarly has donated over $5 million
Oh, wow.
to
The significance of that number is that it's more than the net revenue that's been generated from Grammarly in Russia and Belarus since 2014, which is when Russia illegally claimed Crimea, I think, right?
Yep.
And nobody did anything.
We're also offering Grammarly business for free to Ukrainian nonprofits, NGOs, and media organizations. We're grateful for their heroic efforts. We've added a message in our product to direct users writing about the war to resources for helping Ukraine. We have also blocked users located in Russia and Belarus from using Grammarly products or services. Very cool. So I didn't know that. Thank you for pointing that out, Ken. Didn't know that at all. I used to use another program. It's an email program also made in Ukraine. I can't remember the name of it now. Spark, email program for Mac. All right, one more. David writes, "Hi, Twivers, GAC. Old habits die hard. On TWiV 989, the group lapsed back into repeatedly referring to percent homology rather than percent identity. Yeah, we know that, and I guess we lapse just like we lapse into serum.
Yeah,
instead of
serum and
plasma.
plasma.
If these large proteins are 65% identical, they are certainly homologous. Yes, descended from a common ancestor. You have to talk about percent identity, not percent homology, because they are homologous. By the way, although they couldn't experimentally solve both structures because one protein was not expressed, but with AlphaFold and similar algorithms, they could build reliable models of two proteins that are 65% identical. This would allow them to ask if the structure of key residues in the binding epitope was conserved. P.S. Why are we not seeing new monoclonals being developed? This is a good answer. I like
a
this.
really good question.
It has more to do with expected time on the market than the ability to develop therapeutic mAbs for the various Omicron variants. The UK does better sequence-based surveillance than we do and documented 5 waves driven by 5 different SARS-CoV-2 variants in 2022 alone. Newly emerging variants are presumed to have some level of immune escape. Even if you screen for a new mAb, express it in commercial quantities, get it through regulatory approval in months, which would be a remarkable feat, its commercial life could only be a few months before a new variant which escapes this mAb would likely emerge. To stay in business, a biotech company needs to develop commercially viable products, so failure in months is a problem. Wow. It's a tough business, isn't it?
Yeah.
It is a tough business. But this is way off the subject, but I have to mention this because I ran across it. People in plant diseases are now using monoclonal antibodies made in cell culture to protect the plants. Get that. A mammalian product back into plants, not back into plants, into plants,
Yeah.
and it works.
Okay.
That's remarkable.
What's
Now, how many plant viruses can you think of?
many
Like
different ones.
Gazillion. A monoclonal antibody producer to interfere with plant viral diseases would be remarkable. Absolutely remarkable. Now, remember that mosquitoes, right, Alan? Back me up on this. Mosquitoes feed on plant juices. What if we put human monoclonal antibodies against Plasmodium into plants that the mosquitoes fed on? The next thing you know, you'll interrupt the transmission of malaria through the remarkable technologies that currently exist to us just by mixing and matching, basically. So what's the difference between a plant and an animal? Not much.
All right, time for some pick sticks. And where are we now with the Jazz Project?
Yeah, well, you know, you mentioned that I wasn't picking women much. And the reason why was because back in the 50s and 60s, there weren't too many women jazz aficionados in the jazz field. But there were lots of people, of course. So I picked the ones that I was most familiar with and that I appreciate. But I mentioned 2 of them in this one. So we're now up to the piano. The piano is, in my view, the most remarkable instrument ever invented. It's got 88 keys and you've only got 10 fingers. How the hell do you keep track of all that? And, you know, I've tried to learn how to pick out several tunes on a piano and it just, I I can do it, I guess, but these people are masters. And the master of masters is Art Tatum. If you listen to Art Tatum play, he plays something called a stride piano. His left hand and his right hand, they do know what they're doing, but it's hard to see that they're actually following the same tune because of the different musical rhythms and melding of various themes throughout his playing. Even when he had libs, his left hand and his right hand are doing two remarkably different things. So he's listed by everyone, and I'll follow suit here, as the archetypal jazz pianist that everybody emulates. There was an exception, however, and that's the next one, Thelonious Monk. And I put the S there for a reason, which I'll come back to. This, I won't bore you with all the details of all these pianists, but just know that Thelonious S. Monk, and the S stands for spheroid. He made it up, and he was asked, "Why did you pick that?" He says, "Man, because I'm not a square." And I just love that answer. And he's got offspring that also reflect all of those wonderful jazz attitudes. Thelonious Monk, when you listen to his music, it almost sounds arrhythmic. You're listening, but it's rhythmic. It's highly rhythmic. It's so rhythmic that it's integrated into like 7 different kinds of genomes that it takes to replicate viruses. It's remarkable integration of gray matter expressing itself as creativity. And Thelonious Monk is probably the most creative, inventive pianist that has ever lived. And then I'm not mentioning classical pianists, of course. Dave Brubeck is a standard that I have always loved and one of my favorite early pianists. And Bill Evans is a remarkable, versatile pianist that has transcended 40 years of piano playing and the evolution of jazz. George Shearing never learned how to read music. It's easy to know why, too, because he was born blind. He couldn't read, so he learned how to play it anyway. And he played it remarkably well in every key you can think of. Herbie Hancock and Chick Corea both were pianists with Miles Davis's quintets. Keith Jarrett, who I know you know that too, Vincent, lives in New Jersey, lives not too far away from you, and unfortunately recently suffered a stroke, which ended his piano career. The 2 women that I've mentioned, Diana Krall and Elaine Elias, they're brilliant pianists. And in addition, Diana Krall is a remarkable vocalist as well. Elaine Elias does not sing, but she's a fabulous piano player. Horace Silver is my last pick. And notice I didn't pick just 3 here. I couldn't. I just couldn't. Horace Silver has remarkable music, and you'll recognize a lot of it because you hear it in the supermarkets now. It's so common that everybody now knows who Horace Silver is, except they don't know who was playing. The honorable mentions have to include Oscar Peterson, Errol Garner. Errol Garner, another person, not born blind, but never learned how to read music, can play in every key that you can mention also. Ramsey Lewis, Dr. John. I have to give homage to Dr. John because he studied with the New Orleans outgrowth of Dixieland jazz, a remarkable pianist. He unfortunately passed recently. And Duke Ellington, who was not just a great composer and a great organizer, but also a wonderful pianist. That's my picks.
Cool.
Cone Concert is the best-selling piano recording in history.
Yes, that's Keith Jarrett. And Keith Jarrett is
Amazing.
a remarkable pianist. There used to be a radio show on NPR. Marilyn McPartland was her name. And she had him on as a guest once. And I listened to this religiously. Whenever it was on, I used to listen to it. And Keith Jarrett, she says, now let's play something together, because she was a remarkable pianist also. And Keith Jarrett said, I don't play in public like this. I play when there's something to be played. And when I've booked the gig and I go to the job, I play the job, but I do not play off the cuff. And he left the studio.
Wow.
And she ended up playing by herself, not knowing that was his nature. He was a very circumspect and inwardly directed person who used to announce beforehand, no flashes, no cell phones, and if anybody did one of those two things, he would get up and walk off the stage.
Tough guy.
Temperamental, but a brilliant pianist.
Thank you, Dixon.
You're welcome.
Rich, what do you have for us?
So last week we eulogized Paul Berg,
Yeah.
biochemist at Stanford. And following on that, I got a letter from, an email from a friend of mine who's a graduate student at UT named Jamie Yelland, who reminded me of this video made in 1971 called Protein Synthesis, an Epic on the Cellular Level, which I think has probably been a TWiV pick in the ancient past of
sure.
TWiV. And in fact, if you look ahead in the email queue, one of our other listeners pointed this out as well. But I'm not going to wait. I'm going to go ahead with this. This is a hoot. This was an effort at Stanford University in 1971 in the fairly still early days of protein synthesis, when people were working out all the factors and everything else to illustrate how the genetic code works and how it's translated into proteins, in a video that was done by getting a whole bunch of Stanford students together to choreograph it. Okay, and so it's made partly from an elevated level, and so they form a ribosome, the 50S and the 30S, and they form messenger RNA and codons, and they have tRNA and amino acids and the whole thing. And it's narrated with a bastardized version of the Jabberwocky.
Yes.
That relates how protein synthesis works. And the whole thing is introduced by Paul Berg.
Yeah.
Yes.
Cool.
And man, it is so, actually it's not 70s, it's 60s, okay? Because these are all people.
This is 71. These are people who just had the summer of love, and it shows.
Exactly.
This is
it is just a hoot because it
classic.
is so hippie. It's
Yes.
just great. It's just great. So I
Hey,
recommend
man, I
it.
want to be tryptophan.
There you go. Yeah, that's right.
Not only 71, it's Stanford.
Yeah.
Yeah.
This is so funny. Somebody in the comments wrote, "This is why I laugh every time I hear tRNA."
Yes.
All right.
Because some tRNA must have had an interesting.
Yeah,
Part.
tRNA comes chanting in.
There's a protein. There's a release factor. This lady with the thing jumping up and down and waving, and it's so funny.
And they release a puff of smoke every time there's energy use.
Yeah.
Cool.
Yeah.
Very neat.
Funny.
Yeah, Paul Berg with his skinny tie there. It's pretty
Yep.
funny.
Yep.
That's great. Alan, what do you have for us?
This is my final pick in my series of narrative-driven indie video games. This is Thomas Was Alone. It came out in 2012. I played it just a few years ago. This is, in a sense, it's kind of a traditional video game. It's a platformer. So if you've played or seen the Mario games, famous jumping from platform to platform to get through the level and solve the puzzle. But this one has a story. And I mean, nominally, the Mario games have a story, but it's like really basic. This has an evocative story of a group of individuals who have become aware of their identities and are navigating their way through their world. They are rectangles. So it's just this beautifully minimalistic, and the art in it is gorgeous, but extremely simple. And these rectangular characters, you start to get attached to them as this wonderful voiceover actor gives the narration of what they're doing and what they're experiencing and how they're thinking about it. And they have different personalities that also are related to their different abilities. Some of them have a higher jump or can get through smaller spaces, or one of them can float. So there are all these different characteristics of these characters who you get to know through the course of the story, and you follow along on their adventure. And it's just I think it is the most story out of the fewest elements that I've seen in just about any medium. It's extremely self-contained and just a really wonderful experience.
Cool.
So, Alan, if two of them fall in love, would it be a love triangle?
It would be an extended rectangle at least, yes.
So, Alan, I
did you play?
I bought and played Edith Finch.
Yes.
Okay, this is probably the first video game I've played since River Raid, right? And I thoroughly enjoyed it. So,
Good.
Do you have a favorite of all of these picks?
Gosh, it would be so hard to pick a favorite.
Okay.
What Remains of Edith Finch is a real high watermark.
Yeah, it was really good.
In terms of that. Thomas Was Alone requires more in terms of like reflexes. Some of the puzzles are a little, twitchy, but they're solvable. I'm pretty bad at platformers, and I got through this.
And I have to tell just a little story here. When you mentioned getting attached to characters, it reminded me of the year is 1986. I'm on sabbatical in Boston. I've bought my first home PC, which is a Mac Plus. And my son, who at the time would have been 8 years old, is playing with this, and he's playing Planetfall, which is a story of an astronaut, as I recall, an astronaut who is stranded on a planet, and he's trying to get off. And in the process, he meets and adopts as a sidekick a quirky little robot named Floyd. And we're sitting in our living room, and one evening, my son Jeremy comes into the living room just distraught and in tears and everything else because Floyd has been disassembled. Okay, Floyd is gone, and he was so attached to Floyd, he just couldn't stand it, right? So I didn't know how the game was going to turn out, but I suspected it might, in the end, have a happy ending, okay? So I kept my mouth shut and just sympathized with this, and sure enough, towards the end of the game, Floyd is reassembled and is reunited with this guy, and Jeremy's all excited about it. Crazy.
Yeah.
My pick is an opinion piece in the Los Angeles Times by Michael Warraby, published a couple days ago. It's called, I Called for More Research on the COVID Lab Leak Theory. Here's what I found out. And he goes through how he's addressed this issue. He says, as a scientist who has contributed to several peer-reviewed studies that tell a very different story, I looked on with amazement at the growing divide between what the science shows and what much of the public and a minority of the intelligence community believe. But I've also watched with understanding for those who still suspect a lab leak, because I started there myself. And he talks about how early in the outbreak he thought, well, a lab leak is also possible, so let's look into it. And he began to look into it, and he ended up with finding that the Huanan seafood market was the early epicenter. A lovely paper that we have done here on TWiV, which he says is definitive. It shows that this is a zoonosis. But he says this is how science works. You have to, what did he call it? I have to kick the tires of a hypothesis to try and falsify it. I tabled all my other research to try and falsify the hypothesis that the pandemic began at one of the markets, and he could not falsify it. He said everything stood up. It's really beautifully written.
Not only did everything stand up, but everything he looked at to try to falsify it provided more evidence that it had in fact, and he found new things that people hadn't found before that underscored that this came from the market.
For me, this highlights a particularly annoying thing that I see in some of the columns on this, which is people will drag out the fact that early on some scientists entertained the lab leak hypothesis. Okay, and they'll say, see, these guys even thought it came from a laboratory. But that's how science works. You consider the hypothesis, and these same individuals over time convince themselves that it was not true. So it's totally disingenuous to bring that up as evidence for the lab leak hypothesis. That's evidence for science being done correctly. And the scientists figuring out that no, there's no evidence to support the lab leak thing. And even these guys, and I think even Warby says in this, show me the data, show me the data. You know, I'm not going to tell you it's not possible. No good scientist will do that. Okay, but show me the data, dude.
So he closes it by saying, you know, lab leak proponents cling to the contention that the presence of a lab that studies viruses and the emergence of a corona pandemic in the same city can't possibly be coincidental. But my colleagues and I showed in 2021 that this virus wasn't going to emerge just anywhere in China. It took a city. And, you know, Wuhan and any number of other cities. And he writes, we should instead be asking, what's the chance that a big Chinese city like Wuhan would have a lab doing the kind of research that has come under suspicion? The answer is the vast majority of the biggest cities in China have labs involved in such research. If COVID had emerged in, say, Beijing, there would be no fewer than 4 such labs facing suspicion. I remain open to any and all evidence supporting a laboratory origin. So far, we have no such evidence.
I really like that analysis.
And I like that a lot. And I would just add, if this virus had emerged in New York or Boston or San Francisco or Chicago or Philadelphia or any other major American city, you'd be able to point to a lab probably and say, you know, what are the odds? Well, that's where we put major research facilities.
Yet, the other day at the hearing, the former CDC director said this was made in a laboratory.
He's not a scientist, though. He's not a scientist.
But he's the former CDC director. He shouldn't be saying this stuff when there's zero evidence for it.
No, I know that.
I think there's zero credibility of anybody who was a high-level appointee in the Trump administration.
Yeah, that's exactly what it was.
Any issue.
I mean, that panel was just an engineered attack, right? There's just no. You want to have both sides, right? But there was one side. It's crazy.
No, this whole both sides thing is really annoying when there aren't two sides.
That's right.
Nature has infinite sides.
I mean, this went on for decades with climate change. Oh, we have to cover both sides.
Right.
I mean, I'm in the news industry and I see this. I'm like, no, you don't cover the completely insane side because that's not a side.
Agreed.
False equivalence.
Yeah, false equivalence is exactly the problem.
Yeah, creationist theory falls right into the middle of it.
Right, yes.
We have a listener pick from Carol. It's a book called The Exceptions, Nancy Hopkins, MIT, and the Fight for Women in Science. This excellent book, written by Kate Zernicki, New York Times, previously at the Boston Globe, describes the discrimination against, disrespect for, micro-macro aggressions from the old boys club, marginalization of outstanding, brilliant women in science. The story focuses on the path and career of Nancy Hopkins, but includes other luminaries she interacted with during her professional career. It ends with the acceptance of the Equity Committee report by President Vest that documented wage, space, teaching, institutional support, and other measures which systematically disadvantaged the women on the faculty. Today, the situation has improved dramatically at MIT with more women at every level and in many leadership positions. I remember Nancy Hopkins. Yep. Did you overlap with her at all, Rich?
No.
All right, that is TWiV 991. Show notes are at microbe.tv/TWiV. Send your questions, comments, pics to TWiV@microbe.tv. If you enjoy our work, we would enjoy your financial support. We need it to keep going. You can go to microbe.tv/contribute. Number of different options that you can use there. Dixon Despomier, trichinella.org. livingriver.org. Thank you, Dixon.
Welcome, Vincent. Always, as Richard would say, I won't steal your line, but I had a great time also.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
Love it.
Alan Dove's at alandove.com and theturbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.